how do we measure bioavailability? Well, we can start off with the chemical matter itself, and we can look at chemical parameters. So we can look at things like molecular weight, hydrogen bonding capabilities, log P, the permeability between an oil and a water interface. And this gives you a kind of a very crude idea, but strictly from a chemical perspective. Drug absorption and drug response is a biological phenomenon, so we're better off really getting a more accurate idea if we look at things like the solubility of the drug compound in a water aqueous-based solution like our blood, and the permeability, the ability of the compound to pass through a biological membrane, a lipid bilayer, through a cell, through the other side of the cell, and then into the bloodstream. So permeability becomes very important, and that's really the focus of this lecture, the relationship between permeability and drug absorption. Now there are multiple components to permeability. So there is passive diffusion, and that's in example A, where the compound is simply diffusing through the cell membrane and the cell following its concentration gradient. That's the most simple form of permeability, and many drugs are absorbed by this pathway. There's also active transport, where you have proteins expending energy, moving drugs across the cell and into the bloodstream. You also have paracellular transport, where in this case the drug is passing through tight junctions or gaps that exist between the cells. You can also have efflux pump transport, where in this example E, what happens is that we have proteins expending energy, but they are actively pumping the drug out of the cell as quick as it enters the cell. And then lastly, and least relevant to drug absorption, is pinocytosis. And this is where the drug enters little bubbles in the cell that diffuse across the cell and then pass out that way. So these are the different pathways involved in cell permeability. So is everybody good on that concept? Okay, so experimentally, there are different ways we can study permeability at different levels of complexity. The simplest way, the least complex, but the least physiologically relevant, is to just look at artificial membranes. So we can look at things like liposomes, which are spheres that form around based on lipid bilayers. Essentially, it's sort of like a soap bubble type of thing. And you can measure the transport of drugs from the outside of the sphere or bubble to the inside. Or you can take these artificial membranes and immobilize them on filter supports and just simply measure the flux or transport from one side to the other. These are very simple, but the answers that they provide are also very simple. So you're really better off looking at things like cultured cells. And as I said before, the Keiko-2 cell is kind of like the premier model. This is a cell line, mortalized cell line, that's based on human colon cancer cells. So this is very relevant because it's a living cell, it's human, and it's also from the gastrointestinal tract. So it provides a very good basis for measuring drug absorption of an orally administered drug in a human being. You've got a lot of the fundamental basis built in right there. So these Keiko cells are actually grown in plates that look like this. These culture plates, in this case, this one has 12 individual wells, and the cells are cultured over a period of 21 days in those individual wells. And this is what the cells actually look like. So here on your left is an electron micrograph. This is a cross-sectional view, and you can see up at the top the one side of the cell with these finger-like projections that are very much involved in drug absorption. And the white arrow in this electron micrograph points to a gap junction in between two cells. This is a immunocytochemical micrograph looking down on a monolayer of Keiko cells. So the black areas are the individual cells, and the green stained areas are the gap junctions between the cells. And then this is that same immunocytochemical micrograph, but looking at a cross-section, and you can make out the finger-like projections kind of running along the top of that, where a lot of the gap junctions are occurring. So this is what the cells actually look like. Now in this schematic diagram, what I want to get across is that these cells are very important in all sorts of aspects of drug disposition. So this is a schematic of the cells. You've got the so-called apical membrane, abbreviated by AP, with the finger-like projections running on one side, and then the basal side of the cell running along on the bottom side. And so it turns out what's significant about these cells is not only do they model passive diffusion through the cell, they also model the paracellular diffusion through the gap junctions, but these cells also have many, many different types of protein transporters. They have protein uptake transporters that are involved in the active uptake. They have protein efflux transporters that are involved in the efflux action as well. And so these turn out to be very significant both experimentally and clinically because these cells model very closely pharmacokinetics, the changes in concentration over time due to drug absorption and drug metabolism as well. Because it turns out that inside these cells are also many of the drug metabolizing enzymes that are largely found in the liver. So these are very good models for studying drug pharmacokinetics. They also turn out to be very good models for studying drug-drug interactions. And this is the case which happens very frequently nowadays, where a patient will be getting more than one drug that they have to take at any given time. And sometimes what happens is one drug will monopolize the uptake mechanism that the other drug would use, or the one drug would inhibit the mechanism that the other one uses. And so you get this interference going on, which can either have very toxic consequences or can cause the actual therapeutic dose to be much lower than anticipated. So it turns out that these cells are very good for modeling and predicting those drug-drug interactions. And then lastly, it turns out that cancer cells overexpress these efflux pumps. And so what happens is that you're dosing more and more of the chemotherapeutic agent, but it's not getting into the cancer cell, and it's not killing the cancer cell. And that's because of these efflux pumps pumping the chemotherapy out faster or as fast as it gets in. So it turns out that these can be very useful for studying chemotherapy drug resistance as well. So that's the basic fundamentals behind these Keiko-2 cells. Anybody have any questions about that? All right, then what I'll do is we'll talk about how we use these cells in the laboratory. So in many cases, we're studying multiple drugs, and a lot of times it's useful to use workstations, laboratory robots, to be able to do these experiments. And so you've actually got the culture plates mounted on these workstations. And again, I want to emphasize that these cells are very useful because you're studying transcellular passive diffusion, paracellular diffusion through the gap junctions, carrier-mediated active uptake, and carrier-eliminated active efflux. So if we look at the individual wells in these cultured plates, this is a schematic that shows these. It turns out these are actually two pieces. So we've got the basic culture well on the bottom, and then an insert that sits on top of that. And that actually creates two fluid compartments, the apical compartment up here and the basolateral compartment down here. And then we actually seed the cells right here on this porous support. So we wind up with a cell monolayer with the gap junctions, also called zona occludens, in between, and they're resting upon this porous membrane support. And then we use these experimentally. Here we've got, again, the cross-sectional schematic of the well. The drug is added into the apical compartment. It then gets transported across the cell monolayer, and you can see the apical surface of the cell and then the basolateral surface of the cell that's resting on this support. And the support is typically a polycarbonate porous surface. And then what we're doing is, over time, we're measuring the transport from the apical compartment to the basolateral compartment. So we're measuring the accumulation of the test drug in the basolateral compartment over time. And so we plot out something like this. We're looking at time up to 1.5 hours or 2 hours, and we're measuring the accumulation of the compound over time in the well. And then what we do is we take these data and we can actually calculate a permeability coefficient that's unique for each compound by using a variation of Fick's law of diffusion. So what we're looking at is the change in concentration of the test compound in the basolateral compartment over the change in time, times the volume of the well, times the surface area of the membrane, times the reciprocal of that, times the reciprocal of the original concentration that was added to the apical compartment, and from that we can come up with this permeability coefficient. And what it turns out is these KCO2 cells, which have all of these absorption capabilities, actually turn out to be quite predictive for human drug absorption. So in this plot, we're looking at the permeability coefficients in the KCO2 cells and comparing that with the fraction of an oral drug absorbed in humans in a clinical setting. And so what it turns out is that drugs that are highly absorbed by humans tend to have very high permeability coefficients. And then as the drugs become less and less bioavailable, there's a lower and lower fraction absorbed, the permeability coefficient goes down. So there's the correlation between the permeability coefficients in the KCO cells to the human drug absorption follows this sort of sigmoidal pattern. And so it turns out that these are very useful for predicting how a drug would behave in humans in a clinical setting. So then to summarize, the points that I've covered are that drug absorption relates to drug effectiveness. KCO2 cells can be a very useful and very easy model for studying human drug absorption in the laboratory. KCO2 cells are an easy-to-use lab technique, and they can be used to study bioavailability of drugs. They can be used to study the pharmacokinetics of drug, that is, the changes in concentration versus time. They can be used to predict drug-drug interactions, and they can also be used as a predictor of resistance to cancer chemotherapy. Any questions from anyone?